Okay, I think recording has started. So welcome everyone for the launch of the new Elyra that we have in Liverpool. Just before we start everything, I just let you know that this meeting is recorded. So if you know someone who cannot attend but want to listen later, we can share the recording. I think have decided that you can ask questions after each talk, but you can just post them in the chat. But also if you have a pressing question or you want to say something, you can raise your hand and we will try to check and monitor the chat and the question, and you can ask as many questions as you want. And also what our invited speaker said, that if you need to contact some of the group to get some information on sample preparation or imaging or something, they are happy to be contacted. So that's just for the housekeeping things. Just to introduce a bit this morning, so as probably many of you know, the Liverpool has been successful in a BBSNC 19 alert grant. And thanks to that, we have been able to install the Elyra 7 from DICE back last October, November, and training has just been completed this week, and the installation is under completion as well. So now it will be open for people to use it. So this is why we thought it would be a good time to introduce the system this morning. So as I mentioned, the grant was funded by BBSNC. It was led by myself, Tobias and Daymark, and we had several co-applicants as well, Luning, Natalia, Raphael, Aras, and Dokel. So this is the instrument we have, and you can see, and it will be introduced this morning, that it's bringing more super resolution in Liverpool, and we already had available Eriscan and StreamSurf on our Dragonfly system, but we can now have even more possibilities with the Lattice SIM. And even if you want to go to single molecule localization microscopy, we can do Palmstone. And that will be introduced by Marco and Tom in a minute. So just to introduce the people who will talk this morning, we have Tom Waring, who is covering Jen Adcott, who is our senior technician in the Center for Cell Imaging. And Tom is working part-time with us and finishing his PhD with Toby as well. And he's here to help the user in the CCI training and supporting them. Marie is an image analyst, but also a very good microscopist. And she's been trained, all three people here on that slide have been trained to the new Elyra and can help you. So Marie will tell you about image analysis as well later on. And she's also an expert in light sheet microscopy. And Marco is a facility manager, and he's also expert in light sheet microscopy, 3D imaging, and also confocal microscopy. So if you want to reach out the CCI to do a pilot experiment, access to the instruments, you can just email CCI, and you can check our website, and you can tweet about the event, no problem. I think that's it. Yeah, so the program we have this morning, you will have an introduction by Marco, how to use Elyra by Tom, some image analysis by Marie, and after that we have a double act. In fact, it's written Christoph Baumann here, but we have also Grant Calder, who is here from York, and they had the Elyra installed just a few months ahead of us, and they got it from the same funding stream. So they have already had the opportunity to use it. They have a bit more experience, tiny bit more experience than us, but they already used it. They already have users. They trained 3 users already. So it's a very good. It's very great that we can have some contact with them and get maybe exchange some tips on how to do the training, which happens remotely. All the training the CCI staff had so far was all remote, but it has worked. So yeah, so that's the program for this morning. So I just leave it now to Marco, and I will stop sharing my screen.
Okay, thanks, Jan. Can you all see my screen? Wonderful.
Yeah.
So, thanks for joining. That's great. And good morning to everybody. And so I'm Marco, and what I'm going to do is a very short introduction about the Elyra. So the Elyra is, I like to call it like a multimodal platform, and is actually for fast and gentle 3D super resolution microscopy. What I like about this microscope, from what I could see up to now, is that it is really versatile. So you can either be very fast on the sample if you have fast dynamics, or you can be, if you have fixed sample, you can really push it to the limit of super resolution. I would just like to start with something historical. So the palm technique, which is one part of the techniques on this microscope, is something that is relatively new. And what you see in the picture is the very first palm model that, as you can see, was installed in the living room of someone in US. And this someone was the blonde guy, Harald Hess, in the panel B. And the only reason why it was installed in his living room, it was that the creator of the microscope is Eric Betzig, and won Nobel Prize in 2018, didn't have a garage. And basically, both of the guys were unemployed at that time. That I find incredible. And what basically you will see in the facility is something not much bigger than what was installed in this living room. And now let's see more in the detail how the microscope looks like. So I put here the pictures of two microscopes in the facility. Just to make a point that actually, and just to narrow down and to understand a little bit in broad lines what this microscope is, that's a camera-based microscope. So it's similar to the spinning disc that we already have. So it's not a confocal, it's not a point scanning system. And again, keep it very simple, it's an inverted system. So the objective will be at the bottom, and basically the condenser at the top, and the sample is here. So it's very good to see slide, and very good also to see cell in addition as well. I wanted to include a picture kindly provided by Biomedical of this microscope. Just to explain that it stands a bit off with respect to the average microscope confocal that we have in the facility. So first of all, on the left side, you can see which objective we have. A 20 times is missing. And we have a 10 times, 40 times water collector and oil, and two very high resolution objective. One, the top one is 63 times 1.46. It is mainly used for the turf technique, but it can also be used for other techniques in this microscope. Another thing I wanted to note here is that, as you can see, also the stage is a little bit different from our common stage, which means that not all the support that we use for different kind of plate and stuff that we have in the facility are usable here. I just want to draw your attention on this detail, because actually after the installation and training, I got the idea that this microscope is in a category by itself. So not all the experiments that you used to make in the facility, the other microscopes, are doable in exactly the same form, with exactly the same sample preparation, with exactly the same slide, cover slip, and dish. And that's important, because if you really want to use this microscope at the top, it's also important to prepare the sample like in the best possible way, fitting the technique. And on the right-hand side, you can see the lines, the laser lines that are pretty much standard and present also in other of our microscopes, which is the 405, 488, 561, X642. The laser are stronger, at least 10 times stronger than the laser we have on the other microscopes. And this is, you will understand later, indispensable in order to use the super resolution feature of this microscope. Okay, so the outline of my talk is quite simple. And by the way, sorry, I forgot to say at the beginning, you are welcome to interrupt for a burning question as well. If you have something that is unclear in a slide and you have something you want to ask me straight away. Otherwise, I'm happy to answer also at the end of the talk. So the outline and my talk is very simple. Basically, what can this microscope do for you? And then there would be like a second, also very fast part, citing President Kennedy. What can you do for this microscope? So what is that has to be slightly tweaked and changed in the sample preparation in order to use at best some techniques that are present on this platform. Because indeed, we have to deal here with a multimodal platform. For the CCI users that are familiar with the different microscope in the facility, up to now, maybe the microscope that was able to offer the highest number of techniques was the BioFM, that was able to offer like confocal and wide field and AFM and turf. And this new microscope absolutely beats the BioFM in terms of number of techniques that it is offering, because it's offering SIM, which is structural illumination microscopy, PAM, which is photo-activated localization microscopy, STORM, which is stochastic optical reconstruction microscopy, Apotome, which is, if you want, a variation or a simpler SIM, and TURF, that probably is the technique that is the most familiar for the CCI users. So before going to the single techniques, I would like to address a little bit this triangle that is probably known to most microscopists in the audience, which is the so-called triangle of frustration. The message of this triangle is very, very easy. So basically, you can either have an experiment with something which is extremely fast, with very fast dynamics, so high temporal resolution, or you can push the resolution, the spatial resolution of the picture in order to have the best possible picture, sharpest and the highest amount of spatial information. Or you can try to make an experiment, especially if you have your sample that it is bleaching easily, it is very prone to photo damage. You can try to have low phototoxicity. You can try to have all of these things, but you cannot have the best in one experiment. So you cannot have an experiment super fast with super resolution and low phototoxicity. You have to choose. And basically, what I like about this kind of platform is that you can choose. So you can use different techniques that are the best in each one of these corners of this triangle. So a very quick introduction of the techniques. So Tom and the other speakers will probably speak in more detail about it. SIM is the first technique of this platform that we are going to analyze. It stands for structural illumination microscopy. And we have in particular a variant of SIM, which is called lattice SIM, which means basically an improvement with a lower photon budget, so is even more gentle than the standard SIM, and also faster. But quite simply, what is SIM? So in the panel, what you can see is a sample that is whatever you like, cell or mitochondria or worms or whatever. So this is a yellow blob of sample, and basically what you are doing is illuminating the sample with grid, because what is, if you want the common trait of all these techniques, you don't illuminate all of the sample, you only illuminating portion of the sample, and then you reconstruct the image. And this is exactly what SIM does. So you're projecting the light using a grid or a lattice onto the sample, and you move the grid across the sample, and then you can actually do it with different direction. And what you do by that, you capture multiple images with the sample illuminating different parts, different stripes in this case. And then you reconstruct this series of images and you process them in order to obtain a super resolution image. That's what SIM does. So it is good because you can work with whatever kind of sample, you can work with whatever kind of dye as well, which makes this technique very, very flexible. And then we go to PAM and STORM. And I decided to make only one slide for both of them because they're very similar. For example, one part which is common to both techniques is their single molecule microscopy technique. So basically they are pushing the resolution that you can obtain with our standard microscopy facility to the maximum. But the principle that these techniques are using is a bit different from what is used in confocal. So if you look at the panel, which comes from a paper of Lothar Schermerle, which is one of the guys working in UK with panel super resolution, you have a cell basically with a wide field picture. And the first thing you have to notice for all of these super resolution techniques is that you don't look at a whole cell, and you don't look at a group of cells. You look only at one part of the cell. So as you can see here, we have basically one small square, which is one portion of the cell. And something which is common to this technique, these techniques are always making movies of the cell. Better said, a portion of the cell. So as you can see here, we have a time series where you have in total like 1,000 even more exposures. And you have to think that each one of these dots is a single chromophore. And they work as lamps, so they can be on or off in the different photograms of this movie. So you take this very quick movie, and basically you then will have to reconstruct the image. So there will be some internal algorithms that are taking all these series of pictures, and they are actually, thanks to the individual spot, reconstructing the structure that are present in the cell. So the improvement in resolution you can obtain with this technique, theoretically, is unlimited, because basically you can really see single molecule. Then, of course, there are a lot of factors that come into play. But actually, yeah, you can go really in the nanometer range, like 20, 80 nanometer, depending on the samples. But what you need to do is to reconstruct the image. So you will need some time to acquire the image. You cannot take, like with the confocal, one image in few milliseconds. And then you will have to, thanks to Gaussian fitting, to Monte Carlo simulation, you will have the single points that are building up your image, and you will have a precision and error for the single points. Sorry, I was probably a bit quick about this super resolution technique, but I'm sure that people that are working with it will be able to give more, live, real examples out of it. With the apotome, we take a step back. So it's not as extreme in super resolution as panel store. And if you want, is a simpler variant of the same. So here, what you have is that's basically from the Xi site, actually. You have on a picture of your group of cells, like as a wide field. And then you do exactly the same I was saying for SIM, because that's structure illumination, apotome. That's the principle. So you project a grid, in this case, some stripes, and you take subsequent pictures of your field of view. And basically what you do at the end is you reconstruct the image, and you're able to eliminate the autofocus information. So this technique is faster than the SIM, and is a way of eliminating the autofocus information, similar, if you want, from the point of view of outcome to what the spinning disk does, but different theoretically, because what you do here is not using like smaller pinhole, like with the spinning disk. What you're doing is to reassign mathematically the autofocus light to what is interesting for you on the focal plane. So if you want, is a bit gentler on the sample respect to the spinning disk. But from the point of view of speed and the amount of information that it can deliver, is very similar to our Dragonfly spinning disk. Although, take what I said with a pinch of salt, because we couldn't do yet a comparison like to like with the same sample. Yeah, so apotome can be described as a fast SIM. About turf, I will not spend a lot of time either, because we already have 2 turf systems in the facility, and I'm sure that there is more than one. There are actually several in the audience that already use turf. So I like to define turf as also a super resolution technique, only that the geometry where you have the super resolution is a bit limited. So what you do with turf, you shine a laser light. What you have here is the cover slip, and what you have here are some chromophore. And the physics of it is that you shine the laser light onto your cover slip, and basically the light goes a little bit inside, and then it's just deflected and goes out. But this part of the field of view, which is called the evanescent wave field, is already 200 nanometer, which means that it breaks the diffraction limit of light. So this means that whatever is very close to the cover slip, so if these are chromophores that are very close to the cover slip, like it could be with focal adhesion, for example, with anything that is really close to the membrane, to the bottom of the cover slip, you will be able to see this fluorophore, and you will not see this dark fluorophore. So you could name TURF also super resolution technique because it's very good in eliminating what is out of focus, and it gives a very nice signal to noise. Okay, so now the part of what can you do for this microscope. So at least in my view after the training, but I'm really looking forward to the talks of people that worked with this microscope. What can you do in order to improve the sample preparation pipeline in order to get the best out of this microscope? Yeah, so first impression, actually the training has been completed yet yesterday actually. So basically that's a view of the microscope as I say, like an inverted set of the microscope. And basically the things that we noticed at the beginning is, for example, all these factors that were important in order to get a nice picture in the confocal are even more crucial here. So if you don't pay attention to these parameters, if in the confocal world, in the wide field world, you were getting a bad picture, here you're getting no picture or a picture which is not usable. So in this sense, the few details that I'm going to describe here are really crucial in the super resolution world. So the first one is that the slide has to sit perfectly flat. Okay, and in the standard confocal, we have this kind of holder with two sliders, and once you put a slide there, that's basically done. But we saw also during the induction with the Zeiss engineer following us, that it's very easy to have the slide not sitting perfectly flat. So even if here we have a high precision holder with two thumb screw where you can actually decide exactly the tilting of the slide, is not trivial, even for trained microscopists, to have the slide perfectly flat. But that's important. I'm sure that with a bit of exercise, we will do better. But this fact of having the slide in the green line and not in the orange line is really crucial in order to get a nice picture. What is also crucial is to have a proper camera alignment. If you remember in the first slide, I showed that the system has two cameras, and this camera have to be aligned, especially when you make like a two-color, multicolor experiment. And everything, so stage and everything has to really to be calibrated in order to exploit the full super resolution potential of this microscope. Another thing that is important is basically also the inclination of the incident light. Again, this is something that in the confocal or wide field world may cause slight inhomogeneities of illumination in the field of view. Shadowing in part of your sample here is really compromising your experiment if you don't have the right inclination. So again, the software is great in helping you to have exactly 90 degrees of incident light onto your sample. And there are ways to routine to do it automatically, but that's again one thing where one has to pay attention when doing these experiments. And then maybe the most important point that again is relevant in any kind of microscopy is crucial here is the importance of refractive index matching across the whole light path. So in order to reach this, okay, that's probably the minor point. Whenever you seal your slide is better to have color nail polish because it's helping you to understand exactly where the sample is and where the glue, where the polish is. If you use a transparent one, you might have region that have like a transparent layer of this kind of chemical that used to seal the slide, and then there is like a scattering of the light when it goes through this kind of layer, which we would like to avoid here. Even more important is the cover slip. So only high precision cover slip are accepted onto the system. You can use also the lower one, but then you will have bad pictures, so that's a problem. So only the type 1.5 cover slip with a thickness of 170 micron and a very small tolerance are okay, because basically all the optical path, all the objectives, all the software for reconstructing the images are calibrated onto this parameter. What is also important is the mounting medium. So in the past in the facility, we gave a whole palette of possible medium that you can use to mount your sample, from mobile to vector sheet, etc. Here, apparently, again, if you want to use the system with Storm and reach the highest possible resolution, it appears, as I say, I don't have experience, but Zeiss told us that prolonged glass is the recommended mounting medium because it has the refraction index, which is the closest to what has been taken into account when projecting the microscope. That's the first system for which we will have different immersion oil. So you're already familiar in the facility, there are objectives that want to have water as immersion liquid, something with glycerol, something with oil. In this system, we will use oil mainly, and but we will have oil for different temperatures. So it will be different. The result of the experiment will be really different if you're using room temperature 25, 30, or 37 degrees. And to this purpose, you will have to use the right immersion oil if you're using an immersion objective. You can use this system also with live cells, and the best results we were told by Zeiss, but again, curious to hear what is the experience of people already using it. The best results were obtained with water objective. So in this case, what we have here is an example of a dish with some cells sitting at the bottom of the glass bottom dish. And basically, in this case, you can use oil with the same refraction index of water or water objective, sorry. Water objective is actually apparently the best one to get the best possible image out of your sample. And now I really wanted to, actually, we already had this question speaking with some users even before this launching event about this new microscope. The question is, if I already have a cell line that is labeled with YFP, can I see basically my YFP label cell in super resolution with PAM? And the answer is unfortunately not. So what happens with this actual picture referring to Storm, but the principle is pretty similar. What counts in the super resolution technique is not the chromophore itself or the quantum efficiency of the chromophore or how stable the chromophore is the fact that the chromophore is blinking. So lots of blinking dyes into your picture coupled with high laser power, because those dye are blinking only when they are illuminated with high laser power, then good resolution and a good final picture. Otherwise, you will not have a picture. So that's what I learned during this training, basically. And so basically the question is what makes Palm and Storm different with respect to what we know in standard confocal wide-field microscopy. So you will remember, those of you using the facility, that whenever we are doing induction of the CCI staff, we make a point that the best resolution of the picture is dependent on the numerical aperture and not the magnification of the objective. Now, the numerical aperture of the objective is still important, but another very important parameter, which is not important in standard confocal microscopy, is the number of the photons. Basically, the resolution, so the distance you can see between 2 points becomes smaller, so the resolution becomes higher, the more photons you have. And the mathematical relation is represented basically here, is 1 over the square of the number of photons. And so just to stress this point, because I think probably is the most important take-home message, at least for me, after what I learned in this training session, what is important is to use specific dyes that can be activated and deactivated in order to have this kind of blinking, because the resolution depends on the ability to activate and deactivate at the frequency rate that we want this kind of dyes. And of course, not all the fluorophores can do that. Not all the fluorophores can be photoactivatable. So GFP that is present in many of our ongoing experiments in the facility, the normal GFP is not something you can photoactivate. Photoactivatable GFP is something that can be used for this technique because it goes from a dark state upon illumination with a 4 or 5 nanometer line to a bright state. Then you have some other dyes that are photoconvertible, photoswitchable. When I ask actually during the training, the Zeiss guys, what are the best dyes to recommend to our users, the answer was the Alexa Fluor series. So if you have any antibody with Alexa Fluor, in principle is good, although also in the Alexa Fluor family, there are good and less good dyes. We were told, for example, that Alexa Fluor 647 is the best, easy to use, high blinking rate dyes that having a high blinking rate has a high precision, and then you can get good result. 647 is like a far red line. And basically the message, which is, again, very different from what we are doing in the confocal world, is the more you are on the far red, and the better basically your picture is going to be. The Alexa Fluor dyes in the dye light series in the 500 nanometer range, there are many of them. And what we were told, again, we don't have a lot of experience in the facility, is that they are also good, not as good as the 647, but is also depending on the sample that you want to look at. And the shorter the wavelength, the worse the results. So in the Palm world, ATO 488 is the best of the green dyes, but you cannot rival with what you can get as a resolution nanometer with 647. And that's something really to take into account. So if you have a system, system cells or tissue or whatever, for example, has GFP on it, the right way to try to enter into the Palm Storm world is to couple an antibody with Alexa 647 to the green. So basically, in the Palm world, if you translate everything to 647, in principle, is very good. And that's actually it. So, so thanks a lot. And I'm happy to take a question now, or anytime if you want to drop me a mail, and I will stop sharing now.
Yes, Marco, there is a question from Luning. Thank you very much. Luning, maybe do you want to unmute yourself and ask your question directly? That might be nicer than me reading it. It's about, if you don't, I will ask it. So can we have illumination light around the sample to ensure cell physiology during long-term imaging? Does the growth light need to be off when imaging occurs?
Again, my impression of the training is that the lights should be off because Luning, when you will see the system, you will see that the whole incubation box is totally darkened. And so it's as important as with the multiphoton not to have kind of spurious light into the system. But if you can synchronize the acquisition with the time when the lights are off, I think it is doable.
But does it depend on which modality he uses? Because I assume for Palm and Storm it needs to be completely dark, but for SIM, maybe, I don't know, it's a question. Do you need to have as much darkness for SIM than Palm and Storm?
I would probably turn the question to the people who already used it. My intuition would be that it would be better to have the light off, but please, Christoph.
Christoph has a comment.
Hello. I mean, Grant, please also interrupt me if I say anything wrong, but certainly I'm going to talk about some data from Luke McKinder's lab, who also works on photosynthetic systems, just like I think that's the name what you're asking. And as my understanding is that, I mean, if you build something that's custom, you can have the illumination on to kind of, if you want to, you know, excite your system or synchronize your cells, whatever, to a light source, an external light source. That has to be off during acquisition, but it can be on at other times. So that's the way you would probably have to deal with that.
Yeah, so compared to point scanning confocal, because this is based on wide field technology, I would advise not having it on normal imaging. But after that, yeah, why not? But you have to be able to synchronize.
And obviously, it also helps if you can use specific wavelengths rather than just using a white, white light to illuminate your sample. If there's a certain range of wavelengths, preferably outside the range that would excite your dyes, that would be good too, obviously, to minimize photobleaching.
Okay, cool. Thank you very much for that. Luning, I hope it answered your question, but you can still you can ask more questions to Christoph and Grant if you want. They have a bit more experience than us at the moment. Is there any other question? I don't think so. So we can move on to Tom.
I will share my presentation. Right. Hopefully you should all be able to see that. Okay, so hi, for those who don't know me, I'm Tom, the imaging technician for the CCI, and I'm sort of going to complement some of the things that Marco said by giving us a more of an overview of what the lattice sim in terms of sim and storm palm looks like in practice when actually sort of sat in front of the microscope and what the experiments actually entail. So I'll start with sim because that's probably the easiest. So here we have just a basic schematic of, say, a fluorescently labeled structure in your cell. So you'd mount your sample, you'd find the sample on the dye pieces, and then if you wanted to take a sim experiment, what you'd do is you would project your lattice grid onto the sample. And this lattice grid is then moved across the sample by the microscope during the acquisition. And while this grid moves, the system will take 13 separate wide field images, which is otherwise called phase images. And once you then have these 13 phase images, the software can then process those 13 images into a final super resolution sim image. So to sort of see what these images actually look like, here we've got 3 images. So on the left here, we have one of the 13 phase images that will be captured. And if you actually look closely at the image, you can kind of see a patterning in the signal, which is actually representing the lattice that is being projected onto the sample. Then here in the middle, we have the reconstructed sim image. And then on the right here, we have the sort of reconstructed wide field image. So when you actually produce the sim image, the software can also produce a corresponding wide field image, which is essentially just the 13 phase images averaged. So essentially remove the grid from the image to then give you a sort of unprojected wide field image. So hopefully you can see from these images, which are actually a mitochondrial stain that we just imaged during the course of our training, that we've not only improved the sharpness of our image in order to allow more detail to be seen, but we've also improved the resolution. So this is quite a powerful technique if you want to look at sort of finer structural details sort of within a single cell. And to sort of introduce here a small word of caution to this merry tale. So when we think about sort of confocal microscopy that a lot of us will have all done on a very regular basis, or you mount your sample onto the microscope, you set up your imaging acquisition parameters, and then you capture an image. And so that image that you're seeing on your screen is a direct reflection of the sample and the settings that you use to image it. However, with sim images and also with aperture images that Marco touched on earlier, you've now introduced a step in the middle. So this processing of the 13 phase images into a final image. And so when you're interpreting that final image, you have to bear in mind that what you're seeing is not only a reflection of the sample and the imaging parameters, but is also a reflection of the processing parameters that you've used to generate the final image. This is especially important when you're trying to interpret the image, but especially important when you're trying to compare different conditions, say, within a single experiment, because the processing that you've done will have influenced the final image. So you want to make sure that your processing is consistent across conditions in order to make the maximum comparison power between separate conditions. And so now I'm going to sort of briefly touch on a couple of the more fun modalities. If you're a microscope nerd like me, you'll find these very exciting. So the first one I want to touch on is the time-lapse imaging functionalities. So when we think about what a time-lapse is, a time-lapse is essentially a sequence of images taken one after the other. So when we think about SIM, each image that we get, the SIM image, is actually 13 images that are then processed into a single image. So what that might look like in a sort of traditional sense of a time-lapse is that we might take 13 phase images, which I've sort of represented by these gray boxes here, and we might then process those into our first image. And then we'll take a second set of 13 images and process those into our second image. And so that way we have taken one image after the other in a time lapse. Now, imaging in this way greatly limits the rate at which we can capture our images, because the time between taking one image and the next image is actually equal to the time it takes to capture 13 phase images. So if we want to increase the speed of our sort of time-lapse imaging, what we can use is the burst mode time-lapse imaging. So in this mode, rather than having our sort of blocks of 13 phase images, one after the other, we can instead do a sort of rolling processing of the images. So to sort of give a visual description of what that would look like, here we have our first 13 phase images that you then process into a SIM image. But rather than then taking an entirely separate group of 13 and making that image 2, we instead take one extra phase image and roll 12 of the other phase images into that image, creating a second image. And so again, with the third image, we'd move one along and do the same again. And so this way, by rolling the processing one phase image at a time, we've essentially reduced the time interval between our images from the time it takes to image 13 phase images to the time it takes to image just one phase image, drastically improving the temporal resolution of any time lapse that we capture, while still retaining the high resolution of SIM. So if you're imaging very quick dynamic processes, this method of time-lapse imaging can be very powerful in capturing those sort of fine movements of structures. And now the next one I want to quickly touch on is a mode for 3D imaging. So traditionally, if you wanted to take sort of a three-dimensional image of a structure, what you would do is you would take a stack of two-dimensional images. So you would define a range in height that you want to capture, and then an interval between images that you want to take. And then the system will then capture one image at each point down that range. And you can then take all of those images and recombine them sort of in processing in order to generate a three-dimensional image. Now, with SIM and the lattice SIM capabilities of the Elyra 7, you can instead use what's called leap mode Z stacking. So here you do exactly the same thing in that you define a range in Z or in height that you want to image, and an interval that you want to have between your images. But rather than imaging every single image in that stack, what the system will instead do is it will image every third image, so represented by these black bars here. What you can then do in processing is you can use the out of focus light from the images that you have captured to reconstruct the images that you haven't captured. So in this way, you can essentially capture the same volume with similar resolution in Z, whilst physically imaging less. So this can be especially useful if you have large volumes, since the fact that you have to take less images means that the imaging time is reduced. But it's also useful if you have quite sensitive samples that you want to sort of spare as much laser illumination as possible. This can be a way of minimizing the amount of laser illumination that you give to your sample. Now, of course, as a word of caution, as always with this sort of thing, the processing that you do to reconstruct these images will, of course, influence what you actually see in the final image. So you've got to be very careful to make sure that you process your images correctly in order to accurately reconstruct the images that you're reconstructing. But this can be a very powerful technique if used correctly. And just to sort of touch again with something that Marco mentioned in his presentation, So the Elyra 7 is a dual camera system. So with the filter setups that we have, you can actually image up to 4 colors simultaneously. So when you then think about this combined with burst mode and leap mode imaging, you can do very quick, high resolution imaging with 4 colors simultaneously, which is very exciting for me anyway. So with this in mind, we'll sort of wrap up SIM and move on to what I've termed single molecule localization microscopy, which is essentially just Palm and Storm rolled into one term. So again, we have our sort of fluorescently labeled sample. What we do is we illuminate that sample with a laser. Now, in the case of Palm, this might be a particular wavelength of light that causes a sort of conversion or activation of a fluorescent protein. Or in the case of Storm, this might be a high power illumination to quench a fluorophore, followed by 405 nanometer illumination to cause switching of the fluorophore. But in either case, the practical result of this will be that your fluorophores will blink on and off. You then capture this blinking over the course of a time lapse. Now, this time lapse can be anywhere between a couple of thousand frames and 20, 30,000 frames, depending on the structure that you want to image and the fluorophores that you're using for the experiment. But what the software will do is it will take this time lapse and it will go through it frame by frame, locating each blinking event in each frame, and it will then tabulate all of those coordinates that it's found into one big table. Now, this table can, this can contain thousands upon thousands upon thousands of events. And what this software can then do is it can take this table of thousands of coordinates and reconstruct an image from it. So it will essentially create a new image, putting points of light onto the image where each point represents a previously located fluorophore. Now, to see what this looks like in practice, here on the left, we have an example of a time-lapse image that we took during the course of our training. So as you can see, the fluorophores here are merrily blinking on and off like a bunch of Christmas lights. And here on the right, we have the actual results that you might see from a Storm Palm experiment. So here we have the raw data that is being analyzed. At the bottom here, we have the big old table, which has our X and Y coordinates, as well as a sort of list of other parameters that have been determined about it. And these parameters can then be used to filter the table as well when you're then rendering the image. And here on the right, we have our rendered image. Now, to sort of wrap up with this single molecule localization microscopy by introducing more words of caution. So again, when we think about the final image that we get and how we interpret them, with confocal, you go straight from the system to the image. But with single molecule localization microscopy, you now have not only one, but 2 steps between the microscope and the final image. So the first step being the processing of the time lapse that you've captured of the blinking events in order to localize the fluorophores. And then you have an additional step where you've taken the results that are in the table and filtering those results, removing any outliers, to then produce a final rendered image. And the choices that you make at each point will influence the final image. So when you're planning a Palm Storm experiment, it's very important not only to think about the sample and the actual acquisition settings, but also to think about how you're going to process the images later. So to conclude, structured illumination microscopy, and especially the sort of burst and leap mode functionalities of the Elyra SIM, can allow for fast 2-dimensional and 3-dimensional imaging at high resolutions. Single molecule localization microscopy, Palm and Storm, allow for 2D and 3D imaging at sub 100 nanometer resolution. But that when you're actually conducting these experiments, you must take care when processing and interpreting the images that come out of these experiments. And of course, you're always free to contact the CCI at cci@liverpool.ac.uk. And if anything you've seen here is of particular interest, or if anything you see in these sort of practical uses that we're going to get later from York also look exciting to you, then you can always consult with us and we can see if we can set up some exciting experiments for you. And that brings us to the end of my talk.
Thank you, Tom. That was really clear, really nice. And it was so clear, nobody has posted any question. But well, as Tom said, it's a new technique we have. The CCI will be really happy to do any pilot experiment trial, so please contact us and we'll just try your sample and see what we can get with it. Okay, so I think Marie, you're next on stage. You're muted. Yeah.
Okay. Okay, so I'm going to shift gear a little bit. I am going to talk about the Elyra, but I would like to talk a bit more in general about image analysis at the CCI. We have had a bit of staff turnover since Dave left. I started, then pretty much immediately went on maternity leave. We had a maternity cover, and then now I'm back. So hopefully we should have a bit more stability now in terms of the image analysis side of things. So I would like to give you a bit of an introduction as to why we have image analysis at the CCI, offer this as a service as well as the imaging, and yeah, what we would like to do for you, if possible, and what you can do in the CCI to analyze your images as well. So why am I banging on about image analysis, and why does the CCI have an image analyst? Because in the ecosystem of microscopy, it's actually an integral part of designing your experiment. It can help you make decisions about how you're imaging your experiments or your cells, and then you want to do, or you should do, analysis, ideally quantitative analysis of your images. So it's actually a circle that's where the analysis is part of the whole workflow. It can influence decisions on the experiment, or it should influence decisions, and vice versa. The experiment can influence how to analyze the data. So when I say image analysis is driving experimental decisions, if you wanted to do tracking of cells, and let's say these are two images you've recorded at a certain time interval, and you can see in both images, you can see two blobs. There might be cells, there might be something else that you're interested in, how these things move over time. With these two images, those blobs might well have moved quite a long distance. You don't know whether these have moved sort of in parallel, whether these have actually crossed over, whether these might actually blobs that have come in from outside of the field of view, and the ones that were here to begin with have actually moved out of the field of view. So in this case, you are actually undersampling, and I would say you cannot quantitatively analyze this series of images. So what you want to have is a more appropriate sampling. So as a rule of thumb for time series is the distance between your objects in each image should be larger than the distance an object moves between subsequent frames. And then you're at a point of appropriate sampling. So in this case, there's still a bit of ambiguity, but you'll never get rid of all the ambiguity. So these two objects could actually be crossing paths, or they could collide and then sort of stray from another. But this is a much more appropriate sampling for your experiment. So in this case, the image analysis tells you here, you can't analyze it. Well, you could run a tracking algorithm, but you can't rely on the data afterwards. Here, yes, you can. Just another example. Let's say you have a cell and you have a region of interest where you want to measure the intensity of your fluorophore. In this case, it's a heterogeneous distribution of intensity. So you have some bright blobs and like a darker background, but you actually want to measure all of the various different intensities. So if you were to segment on this particular channel, you might only get the bright blobs, which is not what you're after. You're actually losing information. You're losing the lower intensity information. So ideally what you want to have is an independent channel that labels the same area where your fluorophore of interest is. So you can use this as a segmentation and then apply this mask to your original image and then measure the intensity. So this is another example of image analysis driving the experiment. I appreciate that this is not always possible to do, but if you can design your experiment like that, it is actually much, much better. You'll get much better results, much more reliable results, because these kinds of segmentation algorithms, they always, not always, but often use a thresholding, which means at some point you're cutting off any intensities lower than a certain number, and everything below that is then not measured. So you're actually losing information. So image analysis goals in general is you want to go beyond pretty pictures. Pretty pictures are great. They're winning you image analysis, competitions. But really, for reproducible research, you need more than that. You need good quantitation of your images. So image analysis usually is a multi-step process. You need to pre-process your images. That usually includes some sort of filtering, segmentation, registration, or object tracking. Actually, object tracking is only the preparation for the image analysis. The actual analysis is when you have the numbers that came out of the tracking. And once you've done all these pre-processing steps, then you can measure within your images. You can measure distances, you can measure intensities, you could measure things that happen over time, changes in intensities, changes in localization. Something else you could measure is 3D structures, length of things, volumes. There's, yeah, a lot of things you can do. So you should keep two goals in mind for image analysis. So the first goal is it should allow you to quantify observations. And the second goal is you want ideally to automate your analysis workflow. The sort of the lowest level of automation of analysis workflow is you write down all the things you do at every step of the way. So this is like an analog automation, but you have everything that you need. You have all the information that you need so that you can reproduce this at another time. Another level of automation is to actually have the computer do it for you. But you should keep these two things in mind, not just quantify it, but actually be able to reproduce it one way or another. So there are a couple of image analysis benchmarks. So image analysis is supposed to be quantitative. So you want to derive numbers from your images which describe physical properties of the observed sample. Again, going beyond pretty pictures. The second part, and probably the most difficult part to achieve, is your image analysis is supposed to be objective. Any single person doing anything is never objective. Even 2 people, even 10 people contributing is never fully, truly objective. But as much as possible, you want to get to a point where the derived measurement does not depend on who did the measurement. As in it is free of interpretation. As I said, that's probably the most difficult one to achieve. Your image analysis is supposed to be reliable, so you want to have confidence that your measurement is describing what it is supposed to describe. You want it to be reproducible, so somebody else could do the experiment under different conditions and get similar measurements. And also you want it to be repeatable. So the experiment can be repeated twice under the same conditions and one gets similar results. So these are things that you should always aim for if you analyze your images, and you should analyze your images. So why automate workflows? Well, they usually comprise multiple steps. And in order to have your final workflow, it's an iterative process. So this is just actually a very simple example. You take an image, you denoise it, you do a background subtraction. This is your pre-processed image. So this is already 2 steps. Then you segment your images because you want to measure the size of the blocks or the intensities of these things. You label these and then you do the feature extraction. So this is a very simple pipeline, but it's actually 6 different steps. So you have to remember what you're doing in these 6 different steps. And most of these steps is an iterative process to find out the proper parameters. So there's a lot of information involved here to then retain once you've finalized your process. So workflow documentation, as I said, the most simple is to just write down what are your final parameters for all of these different steps. Is critical for reproducing your results. So if you do the experiment again in 2 weeks, 3 months, a year, or somebody else is trying to reproduce the experiment, they want to be able to do exactly what you've done here as easily as possible. And you need it for writing a thesis or a manuscript for publication. There is a reproducibility crisis in science, and well, not writing down what you're doing here is not going to help with that. And also another point of why to automate workflows is multiple steps add up very quickly. So just the back of the envelope calculation. So if you open the file, it's 1 click of a mouse. Analyze one file. If you include just 1 click per step, denoising, background subtraction, segmentation, labeling, feature extraction, that's 5 clicks and most likely more. So you're already at 6 clicks at that point for one file. Repeat this for all images in one condition. Let's say 10 images per condition, that's 60 clicks. Each condition, let's say a control and 2 different drug concentrations, you're at 180 clicks. So for one very, very simple experiment, you're at a minimum of 180 clicks per experiment, which does not sound appealing whatsoever. If you think that you have a load of images and you need to go through those. So there are actually different ways of automating workflows. There is Fiji batch processing, which gives you a limited number of things you can run with that, but you can still run it over a whole folder of images. If you want to go full on, you can do Fiji scripting and do the whole lot. Then you have all the plugins, all the commands, everything that Fiji has on offer at your fingertips. You can use Cell Profiler, which is actually made for batch processing. It's not made for processing individual images. It is for high throughput image analysis. It was made for things like plate reader data. You can even batch tracking in Amaris. So once you've done your tracking, you can store your parameters for batch and then run it over another time series, given you have used the same parameters on your imaging, the same cells, everything else. So there are always caveats, but it is possible, and it gives you the consistency that you need to then have comparable data. So how can the CCI help you? I appreciate that was a lot of information. So we could help with more simple questions, especially now, given that a lot of people are sitting at home. How do I open microscope images off campus? That's a question that comes up quite often. There are multiple ways of doing that. How do I view the metadata, especially when you're writing a paper or a thesis? What is Fiji and why should I use it? I've mentioned Fiji there. I'm aware you might not be aware of what it is. It's ImageJ with bells and whistles, and yeah, those bells and whistles are really quite handy to use. How do I open images bigger than my computer's RAM, especially for us all working from home? Does my image analysis workflow make sense? Yes, we can help with that. How do I count spots programmatically? For example, you want to count how many cells you have in your field of view and how that changes with drug treatment. Please make the clicking stop and help me write a script, or write a script for me. As I said, 180 upwards. You're in the hundreds, if not thousands of clicks. And many, many more. So whatever question you have, you can always get into contact. So how can the CCI help you? Well, we have an image analyst who can answer these kinds of questions, and I'm more than happy to run you through some of these things if these questions do apply to you. If you have other questions, I'm more than happy to try and answer those as well. But what the CCI offers as well are some powerful image analysis workstations. They are set up that you can access them remotely, and there are 4 workstations. We will have a new one soon, which is more powerful than what we have at the moment. There are different bits of software on there. We can provide training on these softwares, obviously access to these softwares, and support in using them. So the commercial software packages we have are Emaris and ZenBlue. I've also included MATLAB, which obviously you can have from the university, but if you have something that's computationally expensive, running it on a more powerful workstation would help you more. There are a lot of open source software packages like Fiji, Elastic, Cell Profiler, IC. We're more than happy to give advice on these and training. We have the an Elyra server for data management. So there you can have your images, you can view them, organize them, analyze data, make figures for publications, and you can work on your images as well on like a web app or desktop app, or via third-party software, for example, Fiji, Elastic 9, other things. In terms of workflows, we can give you advice on a workflow you've already set up yourself. We can work it out for you. We can automate it with you or for you. Or if there's something that you've never done but you need to do, we can give you advice, or at least help you where to find information. And something that is in the pipeline but not quite possible at the moment with yet another lockdown, children isolating from nursery or school and homeschooling and everything else. But once things calm down a bit more, I would like to set up an image analysis, help desk, coffee break, happy hour. You provide your drinks, you decide what you want to call it, which should be a regular drop-in session where you can ask questions, where we can go through workflows, similar things. So it should be open to people who can drop in, and then if you're happy to share your questions or your workflow advice, then that would be great. Or I could run this is how you do X in Fiji or Elastic or Emaris or something else. But this is to come at some point in the future. So one question is now, how can you help the CCI? So rather than coming to us to say, I have a bunch of images and I want to measure something, I appreciate it's not always possible. We'd prefer if you came to us, I have an experiment in mind, I want to measure X. Let's have a chat about how I can record images suitable for quantitation. So there we're back to the image analysis might actually guide your experiment and your microscope settings, or your image recording settings to make it suitable for analysis. Couple of words on Elyra image analysis. So if you do wide field imaging, you can definitely quantitate spatial relationships, temporal relationships, and intensities. On turf, that also applies. The intensity probably have to qualify. If it's live sampling, things can dip in and out of that optical section, so that it's a bit questionable in terms of the intensity. But if it was a fixed sample, yes, you could probably measure intensities, rely on that. apotome, I would shy away from quantitating intensities because your images are processed. Unless you can image it with a different modality and check it that intensity measurements actually still apply, and the same would be for lattice sim. So, but you can definitely measure or quantify spatial relationships and temporal relationships. On the single molecule localization microscopy, definitely no intensities. And temporal relationships are difficult because they're quite slow techniques. But yeah, the clues in the name, it's all about the spatial relationships on these techniques. And how can you contact the CCI? Well, we've got an email address for those who are at the University of Liverpool. We're all on MS Teams. We all have Twitter accounts. And maybe just one more thing to say is, does a single image analyst have all the answers? No, and I don't pretend to. However, many image analysts have many more answers, and the image.sc forum is a vast repository of information. You can find information over all the packages that I've mentioned earlier, like Fiji, Elastic, Amaris, Cell Profiler, all sorts of things. But also people might have a similar image analysis problem as you have, and you might find some answers there as well. But I'm always more than happy to help. And this is where I'm going to stop rambling, and thank you very much.
Thank you, Marie. I was offline, my broadband dropped off for a few minutes. I've lost the chat. I think there was a question from Anne or Tom. Maybe Grant and Christoph can help about imaging through homemade PDMS dishes. I don't know if you have any view on that from York, if you have tried that.
Yeah, I mean, it just depends on the design. I mean, basically the most critical thing is imaging through appropriate optical glass. So ideally high precision, 0.7 millimeter sort of optical thick glass. You can get away with the standard 0.17, but that's your key thing. Also, I think there's issues about whether the light would reflect off the boundary of the material that you make your wells out of. It just depends on how big your channels are, because you can restrict your imaging. So just crop the camera down or crop the processing down so that it doesn't take that into account when it's doing the analysis. Another point is with when you're doing SIM and apotome processing, it does a contrast stretching, so everything gets brighter. But if you go into the advanced settings, you can set that to rescale to raw. So that would maybe give you something closer to for analyzing intensities. You can also create a synthetic wide field in the advanced functions as well. So which basically doesn't do any contrast stretching. It should just put together effectively a wide field image from all the phases. So there's a couple of options there. So you should be able to quantify intensities. With SIM, well, PAM and STORM, what you can do is you can count molecules. So you're not actually counting intensities as such, but you can do molecule counting. And PAM's particularly good for that because it's basically, you only ever look at one event, so one molecule. Whereas STORM molecule can come in and out of blinking states. So you could image the same molecule multiple times. So you have to be aware of that when you do STORM. You want to quantify intensities or number of molecules. So you're not so much interested in photons, you're interested in number of counts in that case. Number of blinking events you've detected. So does that make sense? So you've got a few options for quantifying.
Thank you.
Another useful thing that I was thinking about as well is with apotome mode is for when you're doing screening, you say take a 96 well plate type format, you know, you can very quickly screen with the apotome mode because you're only doing 5-phase shifts, but still get better quality images than you would with say a wide field system. So this is where the image analysis becomes really critical, because having to analyze 96 wells, maybe multiple positions in a single well, you know, for when you're doing sort of like screening for mutants or drug studies, you know, to do that manually, just, yeah, that would kill you. So this is where, you know, having expertise at hand to do image analysis is like, it's gold. Does anyone got any other questions?
If there are no further questions, probably Violaine is now offline, so I would give the word to the next speaker at any rate, who is, I don't remember, Christoph or Grant or both.
Well, I think it's going to be, I'm going to present.
All right, okay.
And Grant will be the very worthy expert in the background, who probably will take all of the deep technical questions, since he collected, I think, all of this data, which I'm going to present. So hopefully, you guys can see that.
Yes, wonderful.
All right. So, oh yes, thank you for the invitation. My name's Christoph Baumann. So I'm a lecturer in molecular biophysics at the Department of York. And I mean, we've been using single molecule techniques to do both, to look at, you know, do fluorescence localization of biological targets, but also mechanical measurements on biological targets. It's always been the case that we have to build our own kit to make these measurements. So it's we find it a bit of a luxury now that we can essentially stroll up with our appropriately prepared samples and do, you know, this both high res and super res imaging. So it's really going to be a boon for the future. So congratulations to you guys on getting an Elyra 7. You will really enjoy it, and there's, you know, loads of possibility for the future. Grant Calder has also been mentioned. So Grant is our imaging or super res imaging specialist in the imaging and cytometry laboratory, which is located in the Bioscience Technology Facility at York. And that facility is managed by Peter O'Toole, who couldn't be with us today. I'm essentially presenting in his absence. Peter's a very approachable guy, like myself and Grant. So if there's any questions, feel free to, you know, to contact Peter O'Toole if you feel that's useful. So much like yourself, my laser pointer on here. Much like yourself, we have our Elyra 7 system on a pedestal. Showing an image here. And we have, as I understand it, exactly the same system as you guys have in Liverpool. So in today's talk, basically, I'm going to give you 3 courses, and these are just examples of data on bacterial, on yeast, and on algal systems, where we're using some of the one of the standard common modes of super resolution imaging, but also using 2D and 3D sim. Okay, so I'll go into the details of these systems as I progress through. First of all, I'll start with my own work. So one of the things we're interested in is probing the spatial temporal dynamics in the bacterial outer membrane, and we're doing this both using high and super res imaging techniques. So the data I'm going to show you today includes G-storm, specifically 3D G-storm, and some 3D sim images. And the 3D sim data was collected using the standard Z-stack mode. So this is not the LEAP mode, which Tom introduced earlier. And the sample these are fixed E. coli cells. So just a bit of background, just to put in context, so you can see why we're bothering with this. But we published back in 2015 this discovery that in the bacterial outer membrane, so this is in gram-negative cells, that actually you have new protein inserted at mid-cell as the single cell is elongating, so the mother cell is elongating. And the net result is that you end up with a heterogeneous distribution of, if you will, old and new protein in the outer membrane. Okay, so if this is our mother cell, if we label, which we've done, the protein targets in the outer membrane with a fluorophore, and then we grow the cells, and at some point then label the new targets that have been inserted, what we find all the time, regardless of the target, is the new protein is inserted at mid-cell. And I think you can kind of see what could happen here if there's no mixing in the membrane. So basically what happens is you get this partitioning. Okay, and at the single cell level, there's probably not much benefit, but the benefit comes at the population level, because if you're a bacterium and you're switching from, say, a water puddle, muddy water puddle, to nirvana, which would be the gut of a sheep, what you want to do is change your outer membrane proteome, and you can do that at the population level quite quickly if there's no mixing of these proteins in the outer membrane. And you can use this partitioning of proteins and the lack of mixing in the membrane to very quickly develop a population which is completely adapted to this new environment. So in this case, the new environment would be more suited to the protein, for example, labeled in green here. Now, for this to happen, there were two important things that are occurring. One is this asymmetric insertion of new protein into the outer membrane. And we discovered the reason this happens, I'm not going to show you any data on this, but the reason this happens is that the beta barrel assembly machinery, the BAM complex, which is located all over the outer membrane, is actually only active at mid-cell. Okay, so only the machinery at this part of the cell is actually inserting protein. Second important part is on the next slide here, and that is the proteins in the outer membrane don't diffuse. So if you track them, so this is just tracking a monomeric receptor in the outer membrane where we've labeled a protein that binds with high affinity to this target with a fluorophore, in this case, Elyra-488. What we see is that if you track many of these proteins in the outer membrane, plot the mean square displacement in time, this should be linear in time if it's freely diffusing. It is on short time scales, but at longer time scales, you see there's this plateau in the MSC, and that's a fingerprint of confined diffusion. And in fact, if we look at a video of labeled protein in the outer membrane, and this video shows that, so what we've done here is labeled all of the B2B in the outer membrane using curve. What you see, the protein is very much stays where it is, and it also resides in these interesting islands which exist in the outer membrane. So the question is, why is there confinement, and what keeps these proteins from diffusing? Okay, so the other major player in the outer membrane is obviously lipopolysaccharide. Now, lipopolysaccharide is often referred to as just a boring lipid. We don't think so, but you need to be able to see it. And this is another kind of interesting lesson here, is that you don't have to use antibodies labeled with fluorophore to localize targets. Okay, so what we're using here is actually click chemistry, where we're incorporating into the LPS a modified sugar with an azide group. That fortunately, we take advantage of the fact that the KDO sugar is in the head group of this LPS molecule. So this is the part that resides in the membrane. And the fact that this is ubiquitous, this KDO sugar, and you can buy this, feed it to cells, and specifically label your LPS in the cell, okay? And then because this is going to be at the cell surface, we can then use either copper-mediated or copper-free click chemistry to specifically attach alkyne-containing fluorophores to this azide group. Okay, and obviously the azide group is not a common chemical group that you would find in nature, so the specificity is very high. So we did that, and what we saw is that if you look, it's just low-res FRAP data, but basically what it shows is that if you label, at the bottom here, if you label the LPS in a kind of standard E. coli strain, use FRAP, so you're photobleaching this area, what you see is that there's no recovery over 5 minutes. So that tells us that LPS is immobile in the outer membrane, just like the proteins that I talked about a moment ago. If we compare that to in the same cell, looking at the inner membrane, so this is toll A complex labeled with GFP in the inner membrane, that's very mobile. So you get very fast recovery, as you might expect. So LPS is immobile in the outer membrane. Now, moving on to the super res, we want to basically see, is there some organization to the LPS in the outer membrane that's perhaps confining the protein? And to address that, what we've done is use both 3D D-Storm and 3D lattice sim. So if I just start with the bottom here with the D-Storm first, so what we've done here is we've incorporated the modified sugar. We've labeled with Electrofluor 488 dye using copper-mediated click chemistry, and the cells were fixed with 4% paraformaldehyde. And if we look at those cells, this is just basically a Z stack of images. You can see labeling here is this is necessarily optimal level of blinking in these samples, but what you can see is that it's a very heterogeneous distribution, okay, as you move through Z up through the bacterium, okay? And if we look at, this is basically the whole stack showing it where the colors represent the height in Z, you can see that heterogeneity in the membrane. But what's really powerful here is that we actually did 3D lattice sim on the same sample, okay? And here you almost pull out more because you can actually see that there is heterogeneity within the distribution of the LPS in the outer membrane. And we believe that this potentially is the source of the outer membrane protein confinement, because within those kind of holes that appear black here in the outer membrane, those are, we believe, are the protein-rich regions and are the confinement or the cages that keep the protein from diffusing widely in 2 dimensions. And in fact, ongoing work, we're actually labeling the protein in the same way using click chemistry and non-natural amino acids to both do a 2-color experiment that's of what's shown on the slide here. Okay, so the next exemplar comes from the laboratory of Chris McDonald. So Chris is a Sir Henry Dale Research Fellow at the University of York. And one of the things that he's interested in is the study of surface protein trafficking in yeast. And on the exemplar data I'm going to show you, what he's using here is fast 2D sim, image color, a labeled target in these yeast cells. And this basically gives you an example of how fast you can go with this sim technology. Okay. Just once again, a little bit of introductory biology here, just to set the scene. So what Chris's lab does is he has transporters, typically nutrient transporters that have a GFP tag attached to them, and you can add in a nutrient, which is specific for that transporter, that then gets taken up and trafficked to one of the compartments in the cell. So it could be the endosome. So if it's an endosome directed transport or trafficking process, they would have an M-cherry tagged compartment marker for that organelle. They also have Golgi markers, lysosomal markers, and ER markers. Okay, so this is just an exemplar for this particular organelle, the endosome. So in the data that I'm going to show you, it's actually the Golgi that they're looking at. So this just shows you kind of typically what they get with a non-super res approach. So you can very nicely see the cargo at the membrane, and then if you add the substrate in, the nutrient, the transporter then moves from the plasma membrane to, in this case, the endosome, and then later in time, it will ultimately end up in the lysosome. So you get this kind of, with poor temporal resolution, you can see this trafficking process in time. But what the sim actually is enabling Chris's lab to do is to look at this in wide field in real time with sim. Okay, this is 2D, and essentially what you're getting here is you're able to achieve kind of a video which has essentially 1 second frame intervals. Okay, so how do they achieve that? Well, so first of all, the field of view that you're looking at here is 80 microns by 80 microns. So you can very nicely see multiple yeast cells here. As I said, it's 2D sim. So I'm going to show you some snapshots from this particular region, but you can already see in this image that there are things happening at the membrane. Okay, and if you remember from the previous slide, not all of these are essentially not visible in normal microscopies. Okay, so you're able to pick this up in real time. So when Grant did the acquisition here, he used the 2D sim mode, and this basically, in this fast mode, you do lose some information in the Z direction, but what it does allow you to do is run as fast as the system will go. So the limitation here is just the speed of the camera, which in this particular data, the exposure time is 50 milliseconds. And obviously the other limitation is the fluorescence intensity of your sample. You know, is it bright enough? So use then so this burst mode, process the data, okay? And that basically, as I said, achieved a 1 second frame interval in the outputted video. So the target here for the trafficked nutrient transporter was the Golgi. So the M-cherry labeled thing in the background is Golgi, and you can see that moving in and out of the focal plane during the experiment. And so what did this enable them to do? Well, it enabled them to actually see various stages of this trafficking process. So here we see the engagement of the cargo with the Golgi. Okay, you can see that here. Here you see there's this kind of kiss complex at the plasma membrane, because this is where you have the cargo budding off the plasma membrane. You have this running process, and you also have fusion events at the membrane. Okay, so you can look at essentially all stages. You can see all stages of this trafficking process in this set of sim data that was collected in the previous video. So very powerful for following things in real time with very good temporal resolution. And Grant also mentioned that this is very you can also use this in a screening way, because you can very quickly, if you have, for example, a 96 well plate, you can very quickly go across the plate and just, so it's not so much for this necessarily for the super res part, but just for the fast screening of multiple samples arrayed on your slide. And then the last course comes from Luke McKinder's lab. So Luke is a future leaders fellow at the University of York, and his group is interested in carbon dioxide fixation in plants or in photosynthetic cells. And what they were using super resolution light microscopy for is to dissect the structure and function of the algal pyrenoid, and I'll tell you what that is in a minute. And in these experiments, they were using 3D sim. I'll say a bit more about that in a minute. So just once again, just a bit of biology. So our rising global population requires innovation in increase crop yields and provide food security. And one of the interesting, promising ways to do this is to improve the performance of this enzyme complex, Rubisco. And so that's an important enzyme that fixes CO2. And it's actually is energetically wasteful, because there's this process of photorespiration, okay, where you have oxygen competing with CO2 in our atmosphere, okay, and it makes the process of carbon fixation less efficient than it could be. And one way to overcome this problem is to use photosynthetic machinery from single-celled eukaryotes, where they've evolved a biophysical CO2 concentrating mechanism, which enables them to concentrate CO2 around Rubisco. And they do this in this so-called membraneless organelle, referred to as the pyrenoid. And the pyrenoid is an assembly of Rubisco enzyme, and it forms this liquid-liquid phase separated organelle, as it said, which is called the pyrenoid. And these in this particular image here, what we have is in green is the chloroplast. Okay, so this, and later on this part of the single-celled chlamydomonas cells will appear purple colored, so it's the chlorophyll in there that's actually colored when you're doing the imaging. And then in blue, later on in green, is various components of this pyrenoid structure. So the important questions which Luke's lab wants to address is how does this pyrenoid assemble, okay, and divide. It's known that it does divide when the cells divide, and also what are the proteins that make up this pyrenoid. So obviously, if you want to reconstruct this in higher plants to improve the efficiency of higher plants and their output, you need to know how these things happen. So it's a classic spatial biology. And so what Luke's lab has done is they've basically engineered Chlamydomonas to have the protein components that are in actually the entire cell, each of them labeled one at a time with a Venus tag, okay, which is showing up in green in these images. As I said, the purple here is chlorophyll. And this enables them to localize all of the gene products in Chlamydomonas. But of course, what's interesting here is the gene products that exist within the pyrenoid. And they've actually discovered many new components. Thankfully, I'm not for you, I won't be going through all of these. It's this one here, particularly in this panel here, this LT19, that I want to just mention because that's the one that they've imaged using super res techniques. So just on this slide, I'm just giving a bit of a comparison here. So, and this is kind of an evolution of their information that they've acquired using different fluorescence imaging modalities. So using a spinning disc confocal microscope from another manufacturer, they were able to achieve nice images of the pyrenoid, okay, where they've labeled this LC-I9 target within the pyrenoid, and they could generate this model of the organization of this protein within the pyrenoid. Then when we acquired our ARIScan technology in York, we improved on this, okay, and you can now see this kind of cage-like structure emerging, okay, and these imaging, in this imaging they used actually the M neon green tag, okay, and using that same tag with the Elyra system, we're now able to achieve even further enhancements in these images. Just to kind of give you a lay of the land, so this structure here is about 4 microns in diameter. The purple region here is about 10 microns in diameter. Okay, so this is basically 3D sim imaging using the kind of more traditional Z-stack mode for acquisition. And they're in the process of building a model using this data, but it's very much like something that gets kicked around on football pitches. It looks very much like a football in its arrangement. And so basically this SRM data, which they're getting, is going to allow refinement of the pyrenoid spatial model and will guide future experimental work in their lab on the pyrenoid organization. So they're looking at various other proteins which localize the pyrenoid and seeing where they are. And obviously this all we put together to get a spatial picture of how this membraneless organelle is organized. Okay, so just to finish off, so just to know, basically, once again, Grant, who has been involved in all of the data acquisition that I've talked about today. Also noting Peter O'Toole here, who is the head of the Bioscience Technology Facility, and along with myself, we were kind of pushed very much the 19 alert bid, which brought this technology to York. Thank the funding. So obviously DBSOC funded this, but we also had significant contributions from the University of York to purchase the Elyra system for York. And I also want to highlight these researchers from the individual groups whose data I presented today, who actually were involved in collecting the data with Grant. Just want to, before I hand over to the host and take any questions, I just want to say that all of these group leaders, so myself, Chris McDonald, and Luke McKinder, we're all very happy to accept emails from anybody who might have specific questions about how we did things. So please feel free to email us. That's absolutely not a problem. Thank you.
I don't know if Violaine still have problems with the connection. So, well, I would like for the moment just to say.
Back for now.
Sorry.
I leave it to you, Marco, because.
Well, I just wanted to thank all the speakers because we're trying to organize this workshop, and they were really very quick in saying and very enthusiastic about sharing their results, which is something unique. And I would really like to thank them, because otherwise our workshop was less impactful, because it's very nice to have real life cases, basically. I'm also aware that you were staying all beyond the end of this workshop, so it means that the last talk was really, really interesting. Are there any questions? If there are no questions, I probably can start with a very quick one. Grant, Christoph, one point that Zeiss was stressing throughout all the pattern induction, especially when having to do with SIM, was that really this technique are very, very gentle on the sample, the lattice SIM. So can you confirm that? Do you see with the yeast or with the other samples any case of photobleaching, or samples having kind of disruption metabolism or not behaving as they should? Or Zeiss is really right about the fact that the technique is gentle?
So the yeast work was quite challenging. So a lot of it's been started off on our 880 scan system, which has helped a lot with the resolution, but the M-Cherry bleaches particularly badly. So it was quite good that the lattice SIM is gentle enough. It still bleaches. They're very powerful lasers on there, and we're taking a lot of images, but because we can capture 2 images simultaneously with the twin camera system, and we can modulate how fast we go, because then we can really sort of like reduce bleaching down. So with the bleaching, compared to other technologies like soda, it was night and day. It really reduced the bleaching rate down. So for getting high resolution, it was certainly the gentlest that we've done, but there's always room for improvement. So
just to add to that, is one other thing that people need to remember, is that all of those images that you take when you're trying to find things and get the right region of interest where you're going to do the image, they also photobleach the sample. So it really is important to use very low laser powers to find things. And it'd be better just to turn up the camera gain. Use that when you're finding things, and use very, very low laser powers when you're trying to find those. So that's something we found with imaging the bacteria. One thing I should also say I don't think I did say was that you're absolutely right in what you repeated from Zeiss, that you need to carefully match refractive index, etc., to get good images. But I have to say that all of the data that we collected with Grant's help was done without any refractive index matching. It was done with the 63x 1.46 NA oil objective in all instances. So actually that may I say crap data that I showed you today could actually be much better. So we didn't try as hard as we could have to get even better data. So it's possible to do even better.
So, I mean, the nice thing about the lattice pattern is it's more forgiving than the stripe pattern illumination setups. So especially you need that for when you're doing with live cell, because it's the light scatters so much that the pattern breaks down, and no pattern, no super resolution. So it makes a huge difference. I
guess the other point I was going to make is about your comment about dyes. I mean, once Lexiflor dyes, as you mentioned, Marco, are the best ones to use. You mentioned 488 and 647. We also have used 568 with success. So that's another one that can be used. But the really, really important thing here is that you can't just go to your imaging facility and expect that to work. You will have to do some background work to to see which oxygen scavenging system, if you're, for example, if you're going to do destorm, is going to work best for your dyes, both individually and in combination. So we've spent had to spend a lot of time doing that, because where we're doing 2-color imaging of in the outer membrane of LPS and proteins, you know, obviously we need to use different dyes, so we have to come up with an kind of basically a custom oxygen scavenging mix. Typically we use the catalase glucose oxidase mix with MEA present to do the scavenging, but yeah, it's not something you can just look up. You have to optimize it for your system.
And things like labeling density also make a huge difference to how well it's going to work. If you don't have enough labeling density, then it just looks like a disjoint. It's like joining the dots to try and make the structure. But then again, if you've got too much labeling, then you run into the problem of you get multiple spots appearing in one image and you can't separate them. So the system will just ignore them. So really dense areas just become like black holes, because the system will just ignore any overlapping points. So this is just common to storm, not so much the microscope. This is a problem of storm imaging.
Yep.
Other things as well for, you mentioned prolonged glass. Just watch with the prolongs, because when they cure, they shrink in Z, so they compress your sample. So if Z information is important to you, just watch with the prolonged glass, because it'll flatten your sample.
Thanks, that's a really important point.
Yeah. So it looks great, but it's very flat.
Yeah, I should say with the bacterial work that we did, that was all simply done on polylysine coated surface. So we didn't take any. There was no special immobilization of the bacteria there. We were fortunate that they stick well enough that they don't move. So you can get good images.
There's some nice drift correction features built into the Elyra software. So, and the perfect focus system as well for holding the Z position is pretty good.
That's reassuring, thanks. Yeah, so I was having a look at the chat as well, but at the moment I don't see any burning question. I appreciate that you said that you're, Grant and Christoph, you're happy to take question also with mail in the future. So I appreciate the possibility to keep in contact, because we are just starting with it. And I don't know, Violaine, if you want to wrap it up, or I just close it. But feel free.
Yeah, I think yes. Thank you, everyone. Thank you, everyone, for attending. We just hope you will contact us, come with plenty of interesting samples that we can image and play with. Marco and Tom and Mary will be very happy to get more practice. So we need your samples. So please reach out. And thank you all for attending, and thank you, Grant and Christoph, for your input. And that's very much appreciated. I think that's it. We can close the meeting. So thanks.